Good evening, everyone. This is the 11th series of monthly session on the empowerment of Ayurvedic and herbal industries in Kerala. I welcome you all to the session organized by Confederation of Ayurvedic Renaissance Kerala Limited. Ayurveda, the ancient Indian system of medicine, has stood the test of time and is very relevant even in this technologically advanced age. But there are a lot of difficulties or hurdles faced by each element of Ayurveda during the industrial growth. Efforts are being made to tackle all these hurdles. Today's discussion is about the common hurdles faced by Ayurvedic industry. The discussion is about the authentication of mineral drugs and their rectification, challenges faced during the process of manufacturing, documentation issues, and about the common instruments used in quality control lab. The session will be taken by Dr. Jithesh MK, HOD Quality Control Department, Kottakkal Ayurvedashala. Dr. Jithesh is a holder of BAMS from Rajiv Gandhi University of Health Science. Started his career from Kottakkal Ayurvedashala as assistant physician. Later, he went to his postgraduate studies in Rasashastra and Bhaishajya Kalpana from the same university. Handled more than 100 classes ranging from national to international levels in the field of Rasashastra. Credit around 5 articles in various reputed peer-reviewed journals. Currently doing a part-time PhD in Kanjipuram University. I welcome Dr. Jithesh to this webinar. Welcome you, sir, and over to you.
Evam, good evening to all. So hope I'm audible now.
Yes, sir.
Okay. So I would like to extend my sincere thanks, gratitude to the organizers of this CME series. Usually I've been visualizing, I've been observing the topics. Whatever the topics selected in this series were quite interesting. And there is something apart from the something new or apart from the the previous or no, the common webinars conducted by the other groups. Even with Care Keralam, our institution, that Kottakkal Ayurvedashala, has got a very long-standing link connection, and each and every whenever we are in a dilemma, what to do, how to proceed regarding any kind of quality issues or any kind of statutory issues, etc. Our first option is nothing but Care Keralam only. If we need any kind of advanced testing methods, always the first option comes as Care Keralam. So then only if there is something beyond the limit of Care Keralam, then only we think about the other labs, etc. So this is a very, I would like to congratulate the entire institution for doing this venture successfully. So let me once again, I would like to extend my sincere gratitude to Dr. Sharad and everyone, all the organizers. And now let us come to the topics first. So always this hurdles in Ayurvedic industry. Yeah. Okay, right. So this was the topic. All of us, at least those who are not there in the field of pharmacy or any kind of industry, might feel confused. What is this much hurdles in making a decoction or in making a powder or any avalehas or so such kinds? So an outside person, probably he may be a clinician or he may be an academician or whoever it may be, he may feel that, well, is there that much hurdles in an industry? So it's not that the process of manufacturing is not that complicated or complex in comparison with the modern medicine. We don't need to isolate any molecule, or we don't need to do any further kind of the further analysis, etc. And we feel that we can directly, even our statutory regulations allow us to prepare the medicine by ourselves and to use for our own patient, even without a license. So this much freedom we have got in our system. So at least the persons who are standing out from the industry may feel that is there that much hurdles. So I'll just brush up the overall process of industry, as well as the complications what we face in our regular routine work. So now next slide. So hope at least a few years back, probably I have started my education, Ayurvedic education, in the year of 97. So even at that time, 97, see, 97 is not very what is not very far from today's year. It's just 26 years only. So still, at that time also, when I joined my college, many of my friends or other relatives or other neighbors, etc., were asking, so is there something this much to be learned in Ayurveda colleges, etc.? Are you going for a private tuition, or are you sitting with any kind of the old physicians, etc.? So at that time, even just before the 25 years back, the attitude of people were that like that. So there is nothing much to study, and that's Ayurveda. So if the scenario is like that, even the entire Ayurveda is that much easy as per their view. So what complication can happen during this manufacturing? So in almost all these movies, especially in these Malayalam movies, many a time Ayurvedic physicians or Ayurvedic industry, or even some of the patients were also what we say came as, or they have been promoted as a comic characters. So this was the scenario a few years back came in the Ayurvedic in the film industry. So it's a comic. They even one person without any shirt or without any other proper uniform, etc., will be dealing this kind of manufacturing, and the other persons will be just observing that. So that was the scenario previously. So the scenario was just laughing. This was a tool for a comic. So now we'll see next slide. So since I'm not saying that the condition has been changed, since last 5 years, 5 to 10 years, the scenario has been changed a lot. The system has been improved a lot. The government policies has been improved a lot. So, and the entire system became more and more transparent. So now we will see what are the common problems we face in our profession, in our day-to-day profession. So the first one is, all of us knows that it is nothing but the non-availability of genuine raw material. That is the most important hurdle what we are facing now, and we are going to face in the future years. The second one is lack of cost-effective methods to authenticate the purity of raw material. Of course, in the case of herbal raw materials, there are enough mechanical support, the machineries or instrumental support to prove the authentication. There is the liquid chromatography, or even gas chromatography, or even SPTLC. Up to some extent, even TLC can support to identify, to authenticate the herbal raw material. But in case of minerals or metals, absolutely the methods to identify or to authenticate is quite nil. There are some XRD techniques are there, DLS technique is there, but still those are not common and not able, it's not very practical to do or to sample in our daily industry. So we should find out some other alternate methods to find out or to authenticate the the mineral or the metallo-mineral drugs initially. The third problem what we face is the lack of standardization parameters. Only the Ayurvedic Pharmacopoeia of India we consider as our Bible or Quran or Gita or whatever it may be. Okay. So the parameters explained or the monographs explained in those texts in the many times that are beyond our practical wisdom. So maybe such kind of informations or such kind of descriptions might have been evolved from simple academic brilliance, and when we extrapolate them, those kind of informations to a large scale industry, many a time we are moving back. So many a time we feel difficulty to find the quality parameters, to find the consistency in quality in batch to batch. The fourth one is difficulty to obtain consistent parameters. If we take a condition of an arista, if we take this condition of arista, the alcohol content, if the manufacturing was perfect, there can be batch to batch high variations in the alcohol content, or maybe from total TRS or reducing sugar or non-reducing sugar, etc. The value or pH, even pH, maybe the values may be highly fluctuating. And finally, export. When we come to the export, even though there are the rules what we follow in our country is quite customer friendly, as well as this we can say it's a bit loose, but the scenario, the condition is not the same in the importing country. It's quite difficult to import. Even we can export easily, but when it reaches to the importing country, many issues are being happened at any stages, maybe during the time of shipment, during the time of receival, etc. So we will see what are the common problems now. Next. So we'll discuss the first and second 1 and 2 points together, as well as the non-availability of genuine raw material, as well as the absence of the testing parameters or the authentication parameters for the herbomineral, so the metallo-mineral drugs. We know that there are, as I explained previously, there are enough methods, easy methods to authenticate a herbal ingredient. If it is a bala or if it is a guruji, there is a monograph. Even in the API is there, and even in the other library, the monograph library also it's available. So the only one thing what we have to see is we have to take a section of the plant, and we have to check whether it is matching with the monograph kept preserved in the library. If it is matching, definitely the sample is being authenticated, or if it doesn't match, we have to reject. But in the case of mineral drugs, there is no monographs for mercury. There is no monographs for hincula. There is no monographs for this sulfur. So always it's our duty to find a new methods, to develop a new methods to authenticate for this authentication. And in case of mineral drugs, unlike the conditions, unlike the herbal drug, see, if the herbal drug has been mistaken, if we had taken this guluchi, or if we taken patha in place of guluchi for this manufacturing of guluchi adiquata, the only problem what we are going to face is the lack of efficacy. At the same time, if we took naga instead of vanga, or if we took this, if the sample was wrongly interpreted, gairiga as well as hincula, the entire process, the entire product, the entire physical properties of that product is being changed. We cannot get the endpoint reaction. So not only the herbal drugs, the authentication of mineral drugs plays the most important role. And always we cannot depend the the sophisticated instrumental parameters like XRD or DLS, X-ray diffraction or diffractive light spectroscopy. So we need to develop some of the easy titration methods for identifying for the genuineness or the authenticity of the medicine. So in case of herbal drugs, in case of mineral drugs, if the sample what we have taken or was wrong, if it was something other, that can be two options, two problems can be happen. One thing, the health hazards produced due to the wrongly used medicine. The second one, as I explained previously, that we will not get the proper endpoint. The bhasmikarna will not be proper, and the physical parameters will not be proper. Even the chemical parameters also will not be proper, as the chemical structure itself becomes changed completely. So this I wanted to emphasize this once again. The authentication of mineral drugs is quite important than the authentication of herbal drugs. So I will just demonstrate some examples with the help of pictures and how we develop this kind of methods based on the knowledge in the chemistry as well as the knowledge in the field of Ayurveda, in the Rasa Shastra. So these are all just the examples only. And if we going deep into the subject, we can find out much more easier methods. Next. So I will explain just one case. There are total 10 cases I have included this. The diagnosis of the case, the rectification or the treatment of the case. See, after all, we are all physicians. Our main aim is to cure or treat the patient. So just like the other clinical presentations, these are also, as an industrialist, when an industrialist or a quality control person is concerned, these are all disease condition only. So each case by case we will dig. So the first case, we have manufactured one lot of Shankhabhasma. The Shankhabhasma was prepared to the reference from, according to the reference from Dasatarangini. Soon after reaching the market, we have completed the entire procedure. So done the Shodhana with an Nimbusarasa, the Bhavana, the Dola Indra Soyadana with an Nimbusarasa, again triturated and made into villa, made into Chakrigas and subjected for Putta, and again done the calcination. After Putta, it was ground completely. The powder, it was complete, finely powdered, and we sent them to the market. Shankhabhasma as bulk packing, or maybe as 500 mg packing. So soon after reaching to the market, the complaints started raising, stating that severe burning sensation on tongue as well as entire GIT happens during that consumption. Whenever he takes this Shankhabhasma alone, this problem is being increased. At the same time, if he mixes it with a little bit water, absolutely there is no problem. So what could be the diagnosis? What could be the reason and how to rectify this? This is a live problem we encounter. You can see that those who work in the industry definitely will be facing these kind of problems. What is the chemistry behind this? If we know the chemistry behind the manufacturing or behind the process, then only we will be able to rectify that. Whenever if we take a 500 milligram of Shankhabhasma and keep it on the tongue, absolutely at the same point, patient starts severe burning sensation as well as corrosion all the time. So what could be the diagnosis and how we can rectify that? Next. See, we know that actually it's a calcium compound. Both this can happen not only with Shankha, at the same time this can happen with the Pravala also. Both Shankha and Pravala. So we know that both the Sankha and Pravala are calcium compounds. And the calcium compound, after subjecting this calcination, that should be converted completely as calcium carbonate, which is an alkaline substance. At the same time, during the process, there can be some remnants of calcium oxide, which is a corrosive agent inside that. So the entire calcium oxide might not have been converted to calcium carbonate completely. And there will be remainings of this calcium oxide inside the plasma. So definitely what happens then? How to rectify? Why it happens? Why this problem is not happening when when the patient takes this along with water? So the calcium oxide combines with water to H2O and being converted as calcium hydroxide, which is an alkali again. So how can we rectify that? So we have to recall that entire batch, and easily we can just mix some amount of water. We can just pour some amount of water to that entire Shankabhasma, and we can just make that Shankabhasma once again roasted. So the remainings of this calcium oxide will be reacted with the H2O and will be converted as calcium hydroxide. Definitely, which is will be helping as a systemic and as a non-systemic acid again. So if we don't know the chemistry behind the process, we will be in a dilemma. What is the reason for this? We will think that is there any kind of manufacturing defect or any kind of malpractice happens, but nothing. Absolutely, this is a very common issue. It can be happen in any case. So that is the reason you might have seen in this North Indian practice, this Shankha and Pravala as being prescribed as Pishti. This is the reason for making this those to come Pishti. Next. So another case. A case of a lot of Dasamula Haridagi Lehem. We know this, the ingredients of Dasamula Haridagi. So we marketed it. Dasamula Haridagi was being marketed and manufactured. We kept separately. After 1 week or 2 weeks, when we open the bottle or when we when we open the tumbler, we find the fungal growth inside the layer. Even though we add sodium benzoate as the preservative, still the fungal growth being happened. The first time we see, we may think that the amount of sodium benzoate may not be sufficient. So the next lot we add what we do, we will double the amount, and still the problem will be repeating. What could be the reason for this? Why this sodium benzoate is inactive? See, if the fungal growth can happen in any subject only when any substance only when the preservatives become inactive. So what could be the reason for the inactivity of sodium benzoate or benzoic acid or whatever may be the preservative? It may be methyl parabenzodium or propyl parabenzodium or whatever it may be. And what could be the diagnosis of this disease and how can we rectify? Next. So when we see the ingredients of the Dasamula Haridagi, we can see that there is sodium bicarbonate is being added in the name of Java sugar. So always the preservative or the sodium benzoate or benzoic acid, etc., will be acting properly when the pH is at least below 3 to 4. The pH of the product in between 3 to 4, or maybe maximum up to 5. As sodium bicarbonate is highly alkaline, This sodium bicarbonate is the pH, it is making the pH of that product is highly increased. So this was previously the problem was not there, because the sodium benzoate was being added at the time of manufacturing itself, and after some time, maybe after 1 week or 2 weeks, if we add after 1 week or 2 weeks, this problems will not be there. So when the pH increases, the entire substance becomes alkaline, and sodium benzoate in that condition, the kind of this kind of preservatives will be working only at the ideal atmosphere, at ideal pH of 4 to 5. So if the pH is increased, the sodium benzoate becomes neutralized, or benzoic acid becomes neutralizes, and the action of sodium benzoate is being withdrawn. How can we rectify again? So we can add this sodium benzoate before adding the Yavasoga or sodium bicarbonate. So we can just, after this, when the Swangasida has been obtained, we can add this sodium, the preservative, and then after some times, maybe after when it's after 2 or 3 hours is sufficient. Probably after 2 or 3 hours, if we add this sodium bicarbonate, the problems will not be happening. So these are all live problems what we face in our day-to-day industrial life. So the first one was Shankabhasma issue, and the second one was the fungal growth. Third, next. Again, in case of Lohasavam, or it may be Ayaskriti, or any asavas which contains Loha as an ingredient. One lot returned from market because of the reduction in the natural color. So you know that the color of the Lohasava. So at the time of manufacturing, the color was proper. See, these are all imagination, but it can happen at any time in any of an industrialist conditions. So because of the reduction in the natural, the color was being, the dark brown color has been reduced to light brown. And when again 1 month, 1 or 2 months passes, the color was reducing again and again. The color continuously deteriorating as the time passes. So what could be the reason? On iron assay, it was revealed that the iron percentage of the raw materials was considerably less. This is one possibility. I'm not saying that this is the only one possible. These are all imaginary cases, but still, we all can face at any time. Is the reason now we understood? Probably the iron content of the raw material what we used might have been reduced, might have been less. Is there any way to rectify this? Or is there any way to regain this color again? Next. So before that, we had to assay how much percentage iron was reduced. It's quite easy to make this iron assay. You can just see the PPT will be there. You can see that probably the purity, it should be, there is a specific reagent called diphenylamine. So once this stock solution, that the base solution is called as stock solution, and that stock solution is prepared, it has to be reacted with diphenylamine. And if it is the color will be changed properly, and the final color will be the violet. There should be a development of violet precipitate, and the entire color should be the violet. And how can we do this quantification of this iron from this? So there is a calculation. So how much diphenylamine required to reach the endpoint? The endpoint is not the endpoint of this reaction is the generation of a violet color precipitate. So how much drops or how much ml was required to get the endpoint, how much ml of this diphenylamine was required to get the or to reach the endpoint. That amount has been calculated. The amount has been counted, will be counted, and there is a formula for this calculation, and from that we can assess the percentage of iron in the raw material. See, this iron is sparingly soluble in water. So for regaining this color, what happens? It may in the ISPD or Lohasava. Again, if we add simple iron powder or iron patra, will not be enough, will not be sufficient, it will not be useful, as the solubility of iron in water is high. So how can we regain the color again? It's quite easy. This percentage has to be calculated initially, and then there is another iron content. There is another iron compound which is highly soluble in water. It's nothing but ferrous sulfate or annapedi or casisa. So if we add, as per the quantification, the specific amount, if we add, probably maybe 1 gram per one for 500 ml will be sufficient. If we add that, this casisa or this ferrous sulfate is highly soluble in water, and the color will be immediately, within 5 minutes, the color will be regained. So this is how we have to identify the problem, and this is how we have to rectify the problem. Next. Now we know that. So we will just demonstrate some of the what we say, the authentication of mineral drugs as with an example of photos. In the case of Yashada Basma, it is quite easy. Again, Yashada, always we use this Yashada, Naga, Vanga, as well the other Hingula, etc. are the the most common minerals what we use in our industry. So before using this see this Yashada, etc. during this the preparation of Yashada Basma is quite simple. It doesn't have much complicated procedures. How much simple the procedure is, that much authentication or that much authenticity is needed to get the endpoint. If the procedure is quite complex, even though the authenticity of the raw material is little bit less, we can make up in between somewhere. But in case of the preparation of Yashada Basma, the process involved in the preparation is quite easy and doesn't have much complications. And hence, if the Yashada or zinc is not pure, definitely the color will be changed. We will not get the proper this Bhasma, Pariksha, etc. And instead, the final product will be a sticky paste. So always before starting the procedures, before starting this kind of Bhasma preparations, it should be authenticated properly. It's quite easy. Take maybe 1 gram of this Yashada, mix it with one what we say, the 1 ml of concentrated hydrochloric acid, and this Yashada reacts with hydrochloric acid and forms zinc chloride, which is a white color precipitate. That is the end point. If it is quite easy, see it's how easy it is. If such kind of white precipitate has not been obtained, we can understand that there is some mixing. There are some adulteration between. And if we use the same raw material, the final product will not be successful. Next. Again, you might have seen that the product called Trivanga Basma, which contains Naga, Vanga, and Yashada. 3 kind of one. Naga Basma, Vanga Basma, and Yashada Basma. So this color of Vanga Basma, which comes as an ingredient in Trivanga Basma, gets changed frequently. So always see that lead, the purity of the lead is an alarming factor. Although at any cost, lead is the most toxic element what we use in our treatment, and still, if that much toxicity is being there, if the authenticity is also, or if there is some adulteration also happen, what could be the condition of the patient. So always, before using this kind of Naga, Vanga, Hingula, Yesada, etc., Yesada is comparatively safe. This Naga, Vanga, Hingula, etc., you should be the sample should be authenticated properly. So if we don't get the proper color of this Vangavasma frequently, we should check the purity. How? Next slide. Yeah. These are all quite easier titration methods at any lab, even the small scale industries, even the small pharmacies can follow the same kind of preparation, the same kind of methods, which doesn't need any costly reagents or any kind of costly equipments. Only with one thing, we need some 2 or 3 test tubes, 1 or 2 beakers, and some glass ropes, and some one burette as well as 1 pipette. If we could get that, the entire authentication of the minerals has been finished. So it's quite easy. This lead to be powdered and should be mixed with potassium chromate, which is a very cheaper reagent, which leads to lead chromate. It's a yellow precipitate. You can see that the picture is not very clear. That's why there will be some yellow spots inside that reagent. Again, it should be added with the potassium iodide solution. It's a potassium iodide solution is a yellowish or pinkish yellow liquid, which leads to a yellow precipitate, which dissolves on heating with water. If we add water and if we boil it, that yellow precipitate becomes dissolved. And but still, if we subject it to cooling again, the color will be regained once again. So if such kind of reactions happen, we can identify, preliminarily, we can say that the lead is pure. If we need to confirm again with some instrumental parameters, we can go for some ICP OES, or to quantify the percentage of lead, we can go for ICP-MS or ICP OES. Next. Again, Hingula, the most common chemical what we use in our industry. From Vetumaran, we started from Vetumaran or Swasananda, etc. Probably Hingula will be the most common mineral or the chemical what we use in Ayurvedic industry. So how to authenticate? See, this Vetumaran or Chukundipalli or Swasanandam, etc. Even if we are prescribing for young children, the small children also. So the purity should be completely assured of this Hingula, mercury, etc. before starting the procedures. Next. Again, these are all quite easy. It should be powdered completely. It should be mixed with aqua regia. Aqua regia, as we know, it's a mixture of HNO3, that is nitric acid plus hydrochloric acid, in the specific amount, specific ratio. It should be mixed with aqua regia. It should be dried properly. Once it is being dried, it should be mixed with it has to be mixed with the stannous chloride, and which leads to a milky white precipitate. And that milky white precipitate, if we kept for a period of half an hour to 1 hour, that turns to black. So only 2 kinds of reagents are needed for this. Only the one thing is this stannous chloride as well as aqua regia. Out of this, aqua regia is a universal reagent, and we can use broadly in many other kind of reactions and other kind of tests. This stannous chloride is also not very costly, and we can do in our own labs. Only that what we need is some petri dish as well as some of the Bunsen flames. Next. A physician used to prescribe Pravalam, Shankham, Godandi, etc. for osteopenia, and was getting good result previously. But recently, he noticed that the feedback obtained from patients is not satisfactory. So the result was not good. Previously, if the pain, etc., were being reduced within a span of 2 to 3 months, even after 6 months continuous medication, the result is not being satisfactory. So of course, the physician who treats will be in dilemma, will get confused, and he will ask to the quality control people of the pharmacy who is purchased. So again, he filed a complaint against manufacturer. How can the manufacturer convince the customer? These are all preliminary test investigations. Further instrumental, this can be, once the preliminary investigation is being positive, this can be confirmed by the instrumental analysis. Next. So we need to quantify the calcium content of we need to identify the calcium content of any of the calcium containing mineral. It may be Pravala, it may be Shanka, it may be Godanti. So it's quite easy. It has to be ground, it has to be powdered, and simply make it with mix it with concentrated HCl. Take it in a small test tube, dip a glass rod inside the test tube, and just expose to the flame. If we get a brick red color in the flame, that indicates the presence of calcium. And the color varies depending upon the concentration of the calcium. It varies from golden yellow to high red. In case of golden yellow, probably we can quantify as the calcium content will be somewhere around 20 to 40. In case of brick red, it could be some 40 to 80. And in case of dark red, it can be up to 90 or 95. This is how we can quantify the percentage of calcium. Next. So these are all some of the examples, and we can find this kind of easy authentication techniques if we search the textbooks or any other knowledge sources. So what are how to rectify this? These are all individually we have seen that these are the methods to rectify this. Now, altogether, how to clear this kind of problems? Next, the only one thing, the method is to following of GMP, following of QC, quality control measures properly, following of quality assurance method properly. So we will see just what is superficially, what is the quality. Quality is defined as the totality of features and characteristics of the product or service that bears on its ability to satisfy a given need. So whatever may be, the prime aim, it should be to satisfy the indication. If it satisfies the indication, we can say that the method is quite and the material is quite is with high quality. Next. Again, next. Next. So now we will see how or what is the benefit of following this GMP or QA or QC parameters in any small scale pharmacies. It need not be a high range pharmacies, but still even the quality control measures, there should be the quality control wing in any of the small, whether it may be for our own clinical purpose, we should have a small QC lab. If it is there, that helps to maintain the quality. So it helps to evaluation the policy on the scientifically documentation. If we document together completely, we can prevent the recurrence of this kind of problems. Or even if it happens, if it has been documented previously, we can just refer to the previous records and find the solution easily. We should assure the proper storage conditions. We can assure the batch-to-batch variations. That's the main problem. We are not able to, many times we receive complaints that we are not able, the physicians are not able to reproduce the result. If a physician, there may be so many other reasons, If a physician has been given Amritotaram Kashayam to a person and he got result, even if he give the same kind of Amritotaram Kashayam to another person with the same complaints, he will not get the proper result. Of course, we can say that there are the condition, each and every patient are different, and each and every prakriti or dosha or dushya or whatever it may be, and everything is different. But inside that, one element can be the batch to batch variations. Next. 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 Yeah. So we'll see the previously how in the previous eras they were, what were the difference in previous conditions and what is the difference nowadays. We know that previously the medicines were prepared by the physician itself, and it was being prepared by the skilled laborers, and now each and every laborers are being becoming unskillful. In many of the pharmacies, probably we may outsource the laborers, the manual laborers, and everywhere probably can be maybe outsourced. So the skill of the laborer is quite important. And the second one, in the previous era, the quantity were less. And as per the previous conditions, when we extrapolate to a large scale, the conditions are being different. The result is not being proper. So the color of the product we were not able to maintain properly. The taste of the product is not able to maintain properly. And the next one is the seasonal variations. See, the industry which is highly dependent upon the nature is nothing but Ayurveda. If we didn't get proper rain, the entire quality, the entire physical characters of the oils, the thailas or decoctions, etc., is being changed. The qualities of the property, the properties of the medicines, what we manufacture in the winter time, or the rainy season as well as the summer season, may have slight difference as per the. So we can say that the seasonal variations alter the qualities up to some extent. Next. Again, previously, even though the quality control test what we follow nowadays was not being proper, not being there, was not there properly, but still each and every stages were monitored by an experienced Vaidya, and definitely that will be highly beneficial. Might have been beneficial in the previous time. Next. So basically, we can say that quality assurance procedure is the primary responsibility of the drug manufacturer. And that's why QC or QA became kept as a mandatory department of any of the pharmacy. However, the quality of drug should be of concern to a practicing physician. Next. Next. So we know that drug, what is the definition of the drug? The one which we mitigate, which can be mitigated or which can be utilized for any kind of pacification or even for external application. Now even the food substance are also, as mentioned as Ayush Ahar, is being implemented or being incorporated as the government of India and the Ayush department. And there's another variety, the variants now has been evolved. That is Balya, Poshak, Saundarya Prasadak, etc. This cosmetic also has been hijacked or being hijacked by our own ministry now. Next. So we, according to us, Tadeva Yuktam Bhaishadyam Yadarogyaya Kalpadehi. So whatever the things which gives proper health, it can be correlated, can be considered as drug. Next. So now we will see QA and QC just be finished, and now we will just do an overlook over this GMP, good manufacturing practice. What is a GMP? I think this picture clearly explains the purpose of GMP. GMP, we can consider this good manufacturing practice. We can consider as a connection link or as a road from an old kitchen to a newly highly sophisticated pharmacy. The conversion of a kitchen to a pharmacy is called as GMP. Next. See, we can see that uh, usually we can correlate our own industry, this Ayurvedic industry, as a glorified kitchen. Absolutely, there is no other work is being conducted in Ayurvedic industry other than the works which is being carried out, which is being done in the kitchen. Grinding. Previously, it was grinding on the stone, as being converted to grinders. The previous firewood boilers, what we say, the pits, as being converted as the steam boilers or electric boilers. Again, this kind of furnaces has been converted as muffled furnace or maybe overhead furnace. So, absolutely, we can say that Ayurvedic industry can be considered as a glorified kitchen. Next. So, we'll see just the history. Yeah. Next. This was the GMP, the principle. In principle, the GMP was first introduced by NASA in 1962 as the methods to maintain zero defect. Later, this has been brought by WHO, and in India, the GMP in pharmacy company was been started implementing in the year of 1988. Next. Next. So, what are the major domains of GMP that we will just say? One thing is raw material receipt. We have seen that. Then raw material storage. There is a concept called as FIFO, first in, first out. If we don't follow this FIFO concept, definitely that pharmacy will be in trouble within a short time. So the first came raw materials will be kept aside, and the newly purchased raw materials will be using daily, which leads to a high loss of the previous or the damage of the first came raw materials. So always we should stick on the FIFO concept, that is first in, first out. We should monitor the in-process control. There is a record. There should be a record. What's called as BMR, that is batch manufacturing record. Each and every product, whether it may be a small-scale industry or whether it may be a home kitchen for these pharmacies, etc., we should maintain a batch manufacturing record. And I will show the importance of this batch manufacturing records in the next slides. Then the finished goods product quality checkup, storage and dispatch section, product complaint records. Next. So this can be, you may feel that these all can be followed by a large scale industries. How a small industry or small pharmacy like us or like me can implement all this. But definitely, if we don't implement this, if we don't follow this kind of instructions, there will be some, as I explained previously, the batch to batch the variations will be there, the quality deterioration will be there, raw material loss will be there, and finally, the person who is suffering will be the physician only. The absence of standardized documents. That is the main problem in the small scale industries, the small scale pharmacies are being faced. My grandfather will be manufacturing, and my father will be, there will be transferred to my father, and my father transferred to me. Absolutely, there is no written documents. So even if a minimum a raw drug has been changed or might have been misinterpreted, if some mistakes happen, the same mistakes will be repeating generation to generation. Then the another problem is the absence of stock maintenance. The yield cannot be standardized. That's the main problem in the industries what face. If we use 50 kg of Amrutotharan Kashayam, we may get 100 liter this time. Next time, the yield will be 75 liters. And the other time, it can be up to 150 liters. So the reason for this the batch to batch variation in the yield is nothing but the lack of documentation. Then inability to extrapolate to a large scale. If sudden requirement happen, if a person who is in a small pharmacy is manufacturing 50 liters of Amrutotharam Kashayam per month, if he get an order for 1,000 liter of this Kashayam, usually what we should be able to extrapolate the entire mathematics, the entire the quantity to a large scale. We should be able to do that. That many times, if we directly extrapolate, the yield will be less, the color will be less, the physical parameters will be changed. The reason for is nothing but the absence of proper documentation. And again, finally, this is highly individualized. The person who do the proper leha paka, if that person is on that day leave, is on leave on that day, the entire quality will deteriorate. Again, the reason for the absence of proper documentation and the absence of standard operative procedures. Next. So we will just see there are three components mainly for this in the pharmacies. These are all to prevent the further damages, to prevent the batch to batch variation, to prevent the raw material damage, to prevent the in process variations. So raw material, the quality activities of QC activities of raw material, finished product, and process control. Next. So we can say that a set of principles and procedures followed by manufacturer for therapeutic goods helps ensure that the products manufacturer will have the required quality. So this is called as the authentication. Next. Yeah, we will see the raw material standardization. I will show you some of the examples. Next. What are the problems we face now? There are regional changes for the properties. If we take a Bala or if we take Guluchi from what we say, the area of coastal areas like Alapi or somewhere in Kerala, the properties may have entire the change with which is being collected from Wayanad region. Or there may be some change in the properties in Rajasthan region. Again, the properties will be differ if we collect from the Himalayan regions. So this is the regional variation. And the second one is seasonal variation, as I explained previously. This is quite evident in case of shrubs like Brahmi. The color of the Brahmi is differ in what we get in rainy season and summer season. And thereby, the products which is being manufactured, that kind of Brahmi, they also have different physical properties. This is the complaint what we get every day in the industry. Sir, the color of this batch, this particular batch of Brahmi Krita was different from the previous, the batch which I got, the bottle which I received 3 months back. Of course, that can happen. That is the what we say, either we can say that the uniqueness of this Ayurveda, the quality of the raw material or the physical property of the raw material will be entirely changed in the different kinds of season. That's why Acharya specify each season for this collection season for this different kinds of materials. But unfortunately, it's not practical to follow such kind of rules. And the third one is the prohibited drugs. There are different kinds of prohibited drugs like Kushta or any other Datura, or the medicines called as scheduled E drugs, that is Valsanapi or Haridala or Manasila, etc. Gauri Pashana, etc. comes under the scheduled E, which should be taken, which should be used with the proper care. Next. There is a good agricultural practice, GAP, for assuring the authenticity of the raw material, raw herbal material, called as GAP, even though we don't follow much. But for this export purpose, many a times, if we do export to the USFD, US, etc., USFDA is quite stringent for this kind of the following of this kind of GAP. Next. Good agriculture and collection practice, the another one. Previously was GAP, this is GACP. Next. Again, there are guidelines which is being explained in by the WHO for the collection of raw materials. Almost like it's almost in par, it goes in par with the rules explained in Charvidara Samhita. Next. So as I explained previously, I'll just show you some of the SPTLC fingerprint of Valsanapi, which was collected from different areas. You can see the difference in the peaks. The first one, second one, third one. Even though the peaks are similar, the quantity is different. This indicates the difference in the this. I just kept this for making aware of the property change, property variation according to the area where this being collected. So all the components will be there, but their concentration will be different, and thereby there can be minor therapeutic difference also, affecting the therapeutic difference also. Next. See, this is another one for this saffron. So how to assure this? I just put for this to show you how to assure the authenticity of saffron. We know this saffron is quite how costly this saffron is. So just we can extract the active principle. We can get the marker of this active principle, that is crocin, as well as picrocrocin, saffronol, etc. are the alkaloids present in the saffron, and their quantity is being monographed properly. So if we get this saffron properly in the saffron samples, we can make it then an extract, probably the methanol extract, and that methanol extract can be superimposed with the previously done SPTLC fingerprint of the genuine saffron. And if the concentration as well as the peaks are similar, just we can consider it as the genuine one. Otherwise, we can just discard. Next. This is another one in Trivert to authenticate the most common drug in Trivert. We can just get the scopolitan. We can purchase the scopolitan marker, but it is quite costly. But to be frank, such markers are not that much essential for this purpose of this authentication. If we continuously do this SPTLC if we take this kind of fingerprints for maybe for a 10 or 50 samples continuously, and we can get an average value of this, and then when the next sample comes, we can just take this, as I explained previously, the methanol extract or the normal solvent extract we can use, we can prepare, and it can be superimposed with the other one. If it matches with the proper fingerprint, we can receive this. Otherwise, we can reject this. We can accept or reject. We can reject. Next. This is the methods what we can follow in raw material standardization. See, this need not be a TTLC itself. Almost not up to the accuracy is not up to SPTLC, but still we can get some vague idea in the low-cost TLC equipment also. TLC is quite low cost. The only thing is we need a little bit skill to do the TLC. Only that. TLC is thin layer chromatography. Next. Next. So in case of finished goods, there should be the authentication. The foreign matter should be tested properly. The organoleptic parameters, that is the color, taste, smell, etc. should be matching. The LOD should be loss on drying to detect the presence of moist, as well as this ash value and aflatoxin. Very important to further to prevent the further kind of any kind of infections. Next. So there are some still, if it is like this, there are some limitations in this kind of all this test. The one thing is the API has got very small number of monographs and which can be considered as a reference range. There is no comparison. See, we know the kasturi. We can take an example of kasturi. We know that we are not supposed to use this kasturi. So Acharya explained, Bhava Prakasha, Acharya explained that we can use kankola as a bhavadravya of kasturi. But there is no compared data available with the pharmacological properties of this kasturi, as how this kasturi similar becomes with this kankola. Probably the cell line studies or this even animal studies we can use this, but still it should be such data are very limited in number. And there is no much data regarding the regional variations as well as the seasonal variations. So always we should make our own in-house standards for each and every procedures. Next. 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 So we'll go to the standardization of finished goods now. The main issue what we face is non-consistency in result. As I explained previously, Amrudotaram, if we give Amrudotaram to 3 persons with different batch, there can be different result. Up for 2 persons, we will get good result and the other one may not get properly. The reason can be the variations in the samprapti or the variations in the pathogenesis, etc. But we should be kept in our mind that there can be the kind of batch to batch variations also. Deterioration in the quality as per the time passes. So even though we get the shelf life period of 2 years or 3 years, etc., how can we assure the medicine is quite safe as well as it's quite effective till the end of the expiry date, up to the end of the expiry date? So this is the main problem what we face. So there are some trials which we have conducted in our lab. I will just demonstrate it. Yeah, next. So always we should keep, this is called as standard operative procedures, which explain diagrammatic, which it's a diagrammatic explanation, diagrammatic picturization of the entire process. You can see this the purchase of this materials department, then the select from individual compounds, QC checkup with the raw materials as per the API, whether accept or reject, process standardization, from the left line, the water in kind of wet process, steam if needed, oils, the other kind of raw materials if needed, and finally the oil preparation. If is there the patrapagam, the last one is patrapagam if any, and again the process was started with the QC, authentication of raw material, and finally QC checkup with the quality control checkup with the raw material. So each and every product there should be in what we say diagrammatic SOP for the manufacturing. Next. So the product standards. There are 2 books, Ayurvedic Formulary of India and Ayurvedic Pharmacopoeia of India. So there are some monographs which has been explained in both. But according to our own tradition, the medicines included in this API as well as AFI are quite less from our tradition. Most of the drugs included were from this North Indian tradition. And thereby, it's quite difficult to find a testing method or an QC analytical method or a kind of what we say the troubleshooting method in these kind of textbooks. So the main problem what we face is nothing but to make the standardization procedures or this quality control procedures or this troubleshooting methods by our own. So next. Usually these are the tests what we follow. Ingredients, this SOP, probably TLC is the one thing which is being explained in the API. Then the chemical parameters like refractive index, specific gravity, saponification value, iodine value, etc. Depending upon the products. These are all not done for all this product. For some, for example, this kashayam, some other products are some other parameters will be there. In case of Arishtam, the importance will be given to reducing sugar, non-reducing sugar, bricks, etc. In case of Avleham, the importance has been given to LOD, etc. So like that, it varies. Next. So this is the best, even though it's fine, seems to be a little bit primitive. This is the best equipment what we can use for this authentication, this HPTLC, that is high performance thin layer chromatography, which we can use for this identification of the problems, authentication of the rubber raw material, as well as authentication of the finished goods also. If the same molecule, if we couldn't get the same molecule which has been presented in the raw material, and the same molecule is not present in the finished goods, we can find that there's something, some hurdle or some issues is being happened in between the process. So like that, even though we cannot give a report, confirmed report with this HPTLC, which needs for this gas chromatography, etc., for this confirmation, but still we can use as a preliminary data generation method. Next. So you can see this 5 batches of Triphaladi Churna. It's an example. HPTLC fingerprint of 5 batches of Triphaladi Churna. So each and every peaks are similar. Each, and it's called as area under the curve. If the total concentration is similar, and the only difference is the color. That color is being given as per the samples, the superimposition, and there is no significance in the color. So if one peak is missing, there is a peak of this. If we take simple product called Triphala Churna, which contains Haritagi, Amalagi, and Vipitagi. During the manufacture, if a person, during the process, if a person forgot to add this Haritagi, we can just find out through these methods. As we have this peak of this Haritagi, and if that peak is missing in the final product Triphala Churna, we can just say that this is being absent. Next. See, this is the answer for my previous question. How can we assure the quality is not deteriorating till the end of expiry date? So this Kataka Gadiradi Kwatham, the first, you can see this, the first month, second month, third month, and finally the last one is after this, the 12th month, 1 year. The entire peaks are same, the entire concentration is similar, and the entire area which indicates that the properties are also, the pharmacological activity actives are also similar. There is no deterioration from first month to 12th month. This is how we can just get preliminary data with this the HPTLC. Next. See the combination of this Maharasana. This is a comparison of Maharasana the Kashayam as well as Maharasana the Kwadam tablet. You can see there is one another, that violet, you can see some extra peaks. That extra peaks can be either, it can be the preservative used in the tablet, or the binding agent which is being used in the tablet, or as well as in the second one, the first one is the preservatives, the sodium, the peaks can be from sodium and benzoate. Except these two, the entire the properties, the entire pharmacological actives are similar in both these. In the one sample, The final peak is from this paraben sodium or propyl paraben sodium, and the another one, the kashayam, it is sodium benzoate. This is the only difference what we can get. And all the other parameters are quite similar. Next. Now we will see another case. This is a live case. A letter was received. This is quite common, as this drug control department is quite efficient now, and they are continuously watching the entire pharmacy, and the continuous monitoring is being done. And what we say, that we can say this raid itself, the pharmacy is being properly inspected. So a letter was received from drug control department of Ayurveda instructing to show cause for the presence of lead, arsenic, etc. heavy metals higher than the permissible limit in a tablet which does not contain of these metals. Example, Mukkamukurvadi Guliga. We know that in the case of Mukkamukurvadi Guliga, there is no heavy metals are being added. Like there is the Hingula is not an ingredient, or Naga is not an ingredient, Vanga is not an ingredient, arsenic is not an ingredient. But still, if we could find the presence of lead or presence of any other heavy metals in this kind of tablets, definitely it's the responsibility to intimate the manufacturer. It's the responsibility of the drug control department. Sorry, the control department to intimate the manufacturer and to ask for the show cause. So what could be the reason? This problem should not be repeated again. So for that, we should know the reason behind this, how it happened, how this kind of contamination happened in a medicine which doesn't contain. If Hingula is an ingredient in that tablet, if the presence of mercury was beyond the limit, absolutely it's not a damage. If Manasila is an ingredient, if arsenic was found, definitely that also is not an issue. But in case of a tablet which doesn't have any sort of metals, if the traces, even the traces of the heavy metals are being seen, it's an alarming situation. So we should find the exact cause. Then only we will be able to rectify that and prevent from the further occurrence. So what are the reasons for that? That we should find out. One thing is the cross contamination. If the grinding material, the grinder, the previously, if any kind of in that grinder the Swasananda was being out, if the cleaning was not proper, the next medicine charged was Mukkamukurvadi. There is a chance of cross contamination. And if we use in dryers, especially in the hot air dryers, in one tray or in the same dryer, if we keep in one tray this kind of Swasananda or Vetumaran, and in the another tray, if we keep this Mukkamukurvadi, there is a possibility for contamination. If the utensils used for grinding, as well as the filtration, the filters used, etc., can cause this cross contamination. So these are all quite active issues in which is being happened in our industry, and for that we should know, we should be thorough with each and every phases of the procedure. Then only we can prevent this. Next. So the answer is nothing following of GMP. So GMP is for the benefit of the pharmacy people, the manufacturer, as well as the physician. Next. So next, the general requirement, there should be proper separate location with surroundings for the medicines which contains heavy metals and which doesn't contain. It should not be washed together, should not be kept together, should not be ground together. There should be separate grinder for that. There should be separate dryer for the medicines which contains heavy metal, as well as there should be which doesn't contain. The store space should be proper. Working space should be free from contamination. Next. The clothing and sanitation, equipments, and the distribution of records, etc., should be proper. QC parameters. The manager, the physician who is in charge of QC should be monitored each and every phase properly. Next. The location. Acharya clearly explained. Dhanve sadharane deshe same sanmurtige suchau. Smashana should be devoid from Smashana Chaitya Ayatana Shobhaval Mega Varjita. Mrudo Pradakshana Jale, Usharohisha Samskriti, Aphala Kriste, Anakrante, Pataper Velavantara, Sasyate Pesajam Japa. So should be collected from the area where this kind of should be pure, it's pure, devoid of any kind of contamination. Next. The building, there should be adequate light and ventilation, free from insects, cobwebs, etc. If there is furnace, the furnace area should be covered and extra care should be taken. These are all the rules implemented mentioned in the GMP, but we may feel that this for hampering us, the manufacturer. No, but this is for the safety of the manufacturer to prevent further kind of penalty or find a further kind of penalization. Next. Water supply should be pure and potable quality. Washing and cleaning water should be separate. Next. Disposal of waste. Again, whatever the wastes are there, if we throw it away, if the sediments after this, the scrapings of this grinder from the Swasananda, if we thrown it away, again there is a chance of contamination. So the raw material should be kept separately, should be prepared separately, and the waste should be disposed separately. Then only we can assure the safety. Next. Containers cleaning, of course, as I explained. Next. Separation. For the medicines which contains heavy metal as well as which doesn't contain should be kept separately. Volatile materials, something like karpura. If a medicine contains karpura, if we kept in dryer, the medicine with karpura has been kept in dryer along with the other medicine, other kind of tablets. The karpura is the maximum the most volatile one, and the entire gas will be vaporized, and that will be the other medicines also will be contaminated. So we should be give more maximum utmost care to keep away. Next. So the packing material, entire packing material should be kept separately. The rejected material, the complaint material also should be kept separately. Next, these are all just we all know. Always the second one is clothing and sanitation, hygiene of workers. We should insist each and every workers to maintain the quality, as these are all medicines. These are all used by the persons whose immunity is less, who are diseased. So by knowingly, we should not give any kind of harm to any of the persons. So we should the uniform should be hygienically washed. The area should be cleared properly. The hair should be cut properly, and caps should be worn properly. Next. Now we'll see the use of BMR. Next, batch manufacturing record. What is the use? So in that batch manufacturing record, the person who has done the procedure, who has accepted the raw material, who cleaned the raw material, who chopped the raw material, the details will be there, who has completed the procedure, who inspected the name of the physician who inspected the procedure, the person from quality QC cell who inspected the QC quality parameters will be, and everything will be endorsed in that. And if something happens to some violations, some case happened in the court, it is very easy to locate the person who commit that. And for that purpose, this are also the batch manufacturing record also is quite important as per the according to the manufacturer. Next. So always marketing compliance should be recorded. Next. So this is the golden rule of market. No industry or no products will never exist without any complaint. So usually the drug inspectors or this drug controller, etc., comes for this inspection. They should ask to see the complaint register. If absolutely no complaints are being registered, it indicates there are some malpractice. Whatever may be the industry, this is the golden rule. No industry or no products will never exist without any complaint. So getting complaint is not a bad thing, but monitoring the complaint and rectifying the complaint is the thing what we have to follow. Next. Just 2-3 slides for export related and we'll finish that. It's another case. A letter was received from the Customs Department of Germany stating that one shipment of Ayurveda medicine has been seized in airport because of the violation of Washington Convention on International Trade on Endangered Species. On investigation, it was revealed that the medicine was Gugulitha Grada and Kotanjukari Taila. This happens regularly. What is the reason for this? What could be the reason and how to rectify that? Next. The reason was Kotam or Kushta. Kushta or Sasuria lapa or Sasuria costus is a medicine. It's an ingredient which is being considered as endangered. So we need special license from the forest department to manufacture this, the medicines which contains Kushta. And for that, we need to give an affidavit that if we are using this Kushta or Kotta, which has been cultivated by us only. So we can cultivate this Kushta and we can use. We are not supposed to purchase this Kushta or Kotta from any other merchants, and as it's an endangered species. If the main issue happens is this is an endangered species in Kerala or India. So if we have got this certificate that if we are cultivating, we only cultivate this and we use this as an ingredient, that if we could produce that certificate, the clearance will be getting from this our own customs. But at the same time, this may not be sufficient in the importing country. If the importing country doesn't allow at all the medicines which contain this kind of medicines, it may be Kushta or maybe some other ingredients also allowed at all, definitely the entire the entire product will be seized and will be destroyed. This happens quite oftenly. Usually, even the single consignment also, the same thing happens. If some person has been taken treatment in Kerala and while going back to Germany or while going back to US, if he has taken some amount of Grita, Gugulitha Grita or Kotanjukadi Taila, etc., the certificate which I have mentioned previously will be sufficient to get it from Indian airport, but that may not be sufficient to deport from in the foreign countries. So always we should be very careful regarding the issuing this kind of certificates. Next. So what are the issues faced in export usually? One thing is polyherbal products. Our medicines which contains, most of the medicines which contains more than 10 to 15 ingredients. So it's very difficult to find a standard parameters, a standard markers. It's very difficult to find a standard markers. Then different laws to different countries. In the Middle East, the laws will be different. We should be, the labels, etc., should be rearranged according to the rule of the importing country. So each and every country, so there should be separate labels. If we are importing to Middle East, the specifications of that label will be different from the US export, or it will be different from German. So these kind of issues are some big burden or are the main reason for getting proper exporting. Then the demands, the next one. Yeah. Then heavy metal issues. Even though the maximum culprit is this lead, mercury, arsenic, etc., these are the level. Each and every batch we should be able to give certificate. It can be AAS, that is the primitive one, that is atomic absorption. The middle one, ICP-OES, inductively coupled plasma, optical emission spectroscopy. And the most advanced one, ICP-MS, that is inductively coupled plasma. Yeah, so mass spectroscopy. So these are the main tests what we have to follow this. Next. Next. Yeah. So we'll just see the role of physicians in industry to do proper supervision in the face of raw material authentication, raw material finishing. Next. Role of chemist or pharmacist to ensure the proper procedures, to do the QA and the control test, to do the proper record maintenance, to support the physician wherever necessary. Third, next. Role of botanist to authenticate the raw drug, to explore the possibility of abhava dravya, to explore the usage of different parts other than textual reference. We can see this if the root has been in the villa root is been mentioned in the text. If we have to get the root, we have to destroy the plant. So if we can get the same result with the leaf of that root, or the same result with the root and leaf similar, such kind of trials can be conducted. And then, so this is the role of botanist. Next. Yeah. So to get the proper quality of medicine, after all, the medicines are the weapons of each and every physician. Next. To assure the proper stock maintenance, to prevent the seasonal hurdles, to change the individualized dependency, finally to get more profit with less utilization of energy genuinely. Next. Yeah. So this becomes the entire fraternity concern to prevent the further damage from the other systems. We should follow the proper quality manufacturing. We should follow the proper rules and regulations in the manufacturing, etc. So once again, I think I exceeded the time a little bit. Once again, I express my sincere gratitude to the organizers, especially Dr. Sarath and team, for giving me an opportunity. Thank you. So in case of any doubts, I'll be happy to answer if possible. Otherwise, if it is beyond my limited knowledge, I'll just get back to you by the next day.
Thank you, sir, for giving up such a valuable insights on this topic. Now we will go ahead and take some questions. Dear participants, if you have any queries, you may ask, please.
Good evening, sir.
Yeah, good evening.
Thank you for the session. It was very informative.
Thank you.
Sir, I have a doubt. If an aristom is prepared and even after the proper manufacturing, in case of the acidity is increased after a particular time period, so what can be done to what can be done for that particular batch? Even after packing.
Usually the pH should be monitored properly, and if the pH is regularly the pH is increasing, just if we can keep it for some time, and better don't add any other medicines or nothing. The only one thing we can do is we can just mix it with another batch with less quantity, and we can send it together. That's the only method. If the acidity is increased, there is no other way. We may feel that by adding some base like sodium bicarbonate, etc., whether it can be rectified or not. But still, if definitely it's an alcohol preparation, the reactions will be much more dangerous, and it's always better to wait for the watch. And the best method is mixing with another batch.
Whether it will be the new batch will be getting harmful. I mean, it will be spoiled if we are adding like
no, definitely no, that won't happen.
Okay,
that
sir.
will not,
And also,
as this is not a kind of fungal infections, etc., for this fungal growth, etc., that won't contaminate the other one. The only thing will be that they will be diluted properly. Only that thing.
Okay, sir. Sir, one more doubt regarding the aristam itself.
Yeah.
Some of the industries are maintaining a quality that they are reducing the quantity of jaggery so that the alcohol fermentation will not be more.
Yeah.
And some are saying that if you are reducing the alcohol if you are reducing the jaggery content, then only we will attain the proper alcohol percentage. So what is the difference actually?
No, actually, the jaggery content, jaggery is the one thing, the main factor for this fermentation, the helping factor for this fermentation. So there is an optimum amount for this jaggery. So if we have to add, if we say an example of Vasarishta, if we have to add 50 kg of jaggery in the prescribed lot, if the only that 50 kg will be fermented. If the quantity is added by us is 100 kg, or in the prescribed yoga is 100 kg, and the other things will not be, what happened, will be sedimented as well as dissolved as the sugar only. That won't affect the fermentation. So actually, technically speaking, we can just optimize we can take the optimum amount of jaggery and we can add to that, and that will give you good fermentation. But the problem is that is against the rule. That's against the that's violation of the pharmacopoeia rule. As in a yoga, if it is a PNP yoga, patent proprietary yoga, you can do like that. But if it is a classical yoga, you are not supposed to change the ratio, as well as you are not supposed to change the concentration of any of the ingredients. So
sir, in that case, that alcohol percentage will be more, no, sir? If the fermentation in jaggery amount is more.
The alcohol percentage should be optimum. It should not be more.
Okay.
That's the one thing. So for that purpose, the best thing what we can do, if it is a patented preparation, you can just find out the optimum quantity of the sugar or jaggery what need for this fermentation, and you can add that amount itself. Absolutely, there is no need of adding extra. But if it is a classical one, if 100 kg is explained, you have to add the 100 kg itself. Otherwise, that's a violation of rule, isn't it?
Yes, sir. Thank you, sir.
Hello, I'm Dr. Kiran. Can I ask a question?
Yeah, sure, sir.
Yeah, okay. Dr. Jithesh, I'm basically from Germany.
Right.
So we are aware of all these issues, basically the CITES and all these kind of things.
Right. Yes, yes.
So if we follow that, then actually it's like that almost 95% of the Ayurvedic herbs cannot come to the European Union. If we go
exactly, it's not possible. Yeah.
So then we have another issues like the big manufacturers are not really attending this issue because they don't have a much market here. So they all try to bypass the law, including Himalaya or Charak, and also they use other countries to bring the product. That's one thing. But my question is basically about the standardization practice which we have here is basically representing the product, a polyherbal product as it is. That means once you use a standard, and then we are supposed to maintain the same standard all the time. So
exactly, that's the main. That's what I mentioned. The assembly.
So here we have serious issue like that. We both clearly know very well that Guduji or the Rasta, Guduji is available. Rasta and Bella, which are the main ingredients, that is not used as the single species. That is used as per the API. Yeah? So it may not be the Sida rectusa when you are getting the same time. So I think none of the Ayurvedic manufacturers are in a position to use Sida in that quantity, because we have in Kerala itself more than 600 manufacturers.
Yeah, right.
So all the different, I assume now they are using Abitulum indicum, and then now we have the same thing with this Rasna. We have this Pluchia
lanceolata and Alpinia galanjara,
and all these kind of things are coming. And then the second quality of Alpinia cacarata is, I don't know, it's a kind of costus ginger, which in local name is Malainji, which is widely cultivated in our high range areas. So
It just got a
yes,
same flavor.
yeah, so that is the second quality of the rasna, which has nothing to do with the rasna at all, except the
Exactly.
smell.
Yes.
So probably the rasna churram is the only one product in the market which has the smell of which resembles rasna through this melange. So the including the same issue is also there with this ashoka. So this is what we facing, like that when we represent one product in its entirety, and we are in a no position to replicate the same same product. So what do you think that what could be the major medicinal herbs which already extincted or which is not really available in the industry? Because I think you are coming from the QNC. I have some experience. I was the operation head for Patanjali in USA, and I've been with Nagarjuna also. So I have seen that they use more than 1,500 mix for 1 year production. So I have never seen that any manufacturer in Kerala is complaining that we don't have this particular raw material, so we are not producing this particular item. They never happened. So I was surprising. Maybe in 20 years' time, can you make a prediction that what could be the species which may not be available?
To be frank, we'll be getting, if the situation goes like this, almost 50% of the raw materials will be extinct.
Okay, thank you. Yeah.
That's the fact. So every year, we feel much difficulty to procure. The difficulty in procurement is being increasing every year. That's the main problem. That's why there's this one Ashoka, Villua, and everything. These are all whatever the entire things in the Brihat Panchamula. Brihat Panchamula is almost, it's very difficult to get. Then this Panchavalkala, it's not at all available now. Sir. Only three or four are there. So even we feel scarcity even to obtain Shatavari also. So probably it's a very alarming issue. And in case of minerals, there is in case of herbal drugs, there is a possibility that we can cultivate. But in case of minerals, if it has been used completely, absolutely that will be ended for full. What is that? That will be the end of the day. We cannot even prepare synthetically.
Doctor,
So,
actually, as long as we don't admit that we have this problem, then it is not a problem. So
definitely,
like that, nobody in Kerala will admit that the Bella or the Sida rectusa or cordifolia is an extincted species. So that is one thing. So
I think is we'll be getting all the species occasionally. Probably will be mixed one. That's the main problem. All that raw material what we're getting may not be abutyl and indigon also. They will be mixture of Sida rectusa or Sida cordifolia. Probably some 10% will be there, and this happened in all the cases. So we cannot say that completely extincted. The availability is quite less, but if the situation continues like this, it's going to be very pathetic. Need to do something, and there comes the importance of single herb formulations as well as the formulations with less number of medicines, rather than using this kind of Chavanaprasam or Mahanarayana Taila or Mahamanjishtadipatha, etc., which contains more than 35 ingredients. So always we should try to substitute. We should learn to substitute with the small group of medicines. This is the only one thing what we can do.
Doctor, just one more question. Is there any studies done in your unit for to find out the damages caused by the preservatives actually? Because recently when we evaluate the claims or the accusations against Ayurveda, especially going through Dr. AB studies and all, we found that they are talking about a particular parenchymal damage of the liver tissues. So they use more than 100 species, but only one type of damage they could find and identified in this entire group. And probably we assume that that's coming from the preservatives, because it's when you take 100 drugs, you cannot identify and study the 100 drugs for any liver damage, but all the common factor in these entire drugs was these preservatives, especially the one from Patanjali is Guduji Sattva, or the Kamilari and all, which is commonly people use as an OTC market. So is there is any study, Kottakkal is being a leading institution in Ayurveda, there was any studies done in your unit for these preservatives? Because use of preservatives, Kottakkal, I assume they use a lot of preservatives because the volume you produce and
Yes,
so the liquids
exactly.
and all. So Kerala is one of our industry may be using more preservatives than the entire Coca-Cola in the country. So because the same quality of preservative, quantity of preservatives required in the food industry is about 6 months, and the same preservative when we could use it for about close to 3 years. So the loss of preservatives are same, no? And even is there is anything we have? This is also one thing we face in Europe, basically the issue of preservatives.
No, there is no published literature around there, anything. But the only thing, I should be proved through either animal study or at least with the clinical study. Cell line studies or something will not be, or the chemical studies, chemical profiling, etc., will not be sufficient. That which is a very strenuous procedure, and till now we didn't conduct such kind of studies.
Okay, thank you, doctor. Thank you for being honest. Thank you.
Good evening, sir.
Yeah. Hello.
Sir, I have a doubt. I am a part of a pharmaceutical company in the
Right.
QC section. We face a main problem in decimal refining, crystallization in decimal refining. What is the cause? Can you please explain?
I couldn't get you, madam.
Crystallize.
Can you? Was not clear.
Crystallization in decimal refining.
Crystallization in decimal rasayanam in entire batch.
Most probably in different batch it occurs. We reduce the sugar content and we change the preservative composition. Now it's almost okay, but it is the preservative same for all other preparation. But in the Decimula rasayanam, the major problem is crystallization.
Okay. That has to be. Just need to see the ingredient list completely. I don't remember the complete ingredient list. The most common crystallization usually happens in Draksharishta or Mrudyugarishta, even Drakshadi Lehya, etc. Or probably not in Lehya. The Draksharishta, Mrudyugarishta, etc. is quite common. As this, it's a this kind of crystals can be. It's called as calcium tartarate, which is a byproduct of wine industry. Usually it's in the wine industry, it is quite common. Being this procedure is the same, the medicines which contain draksha, this grapes usually being converted as this kind of crystals. It's a typical glass-like crystals, and which is known as calcium tartarate. That is the most common crystallization what we have seen in kind of decimal rasayanam. Let me inquire. Let me. I will get back.
We feel it is sugar crystals. We are adding camphor in that, but we feel that it is sugar crystal.
No, camphor actually is a volatile one. That one produces this kind of crystallization.
It is not camphor.
That one lead to crystallization. So probably something else. What preservative you are adding? Is it benzoic acid or sodium benzoate?
Sodium benzoate. Sodium benzoate plus a combination of SMP and SPD.
Okay. What concentration?
That I have to check.
Okay. Anyway, I'll get back to you. So anyway, you can, if needed, I'll just write my. I'll tell you. I'll do my number. So you can just put it down. My number is 9447. 9447. 92 5657. I'll write it. 9447 925657.
9447
925657. Yeah, I have typed in the chat box.
925657. Okay.
Yes. So I'll get back to you. Let me check.
Okay, sir. Thank you.
This is the calcium tartarate is quite common. which is in the Draksharishta, this kind of aristas which contain grapes.
Ah.
Even Brahmi Drakshadi Kashaya, etc. also happens. Even Drakshadi Kashaya also is quite common. But not in Lehas, it's seen.
Okay, thank you, sir. I will contact you.
Yeah, sure.
Sorry to interrupt, sir. One more doubt.
Oh, sure, madam.
Sir, while packaging of the Pinda Thaila in 450 ml bottles and all, after a particular time period, the shrinkage of the bottle has been seen many a times. So, what can be the reason for that?
Probably the one thing is the what we say, the air pressure generated due to the compressed filling. That's the most common one. So nothing else. In aristas, it's quite common because of this fermentation. But in this kind of thylas, the pinda thyla or the andhra thyla, etc., usually it's not very common. The reason could be the excessive air pressure. And you can do you leave gap between or do you fill completely?
We have we are not filling it completely, sir. To the apt amount only we are and there is spaces, air spaces also there. But even if we are loosening the cap, we are not getting any air. Air pressure like that, sound and all are not seen at all.
Not seen. Okay, you can just do one thing. You can just do a prick on that any of that area of that the top of that bottle. Just as a single needle prick. That's how we differentiate here. If something happens, just we do a single prick. If it is due to this kind of air pressure, just that will be relieved by that. And this is quite common in this honey, etc. So, so usually this happens, but still the other thing could be just we have to
Mainly seen in Pindathylam only, sir. Not in any other Thailam or Colombo. We are not seeing that.
Okay.
Whenever any batch we are preparing the
And
Pindathylam.
it's filled only after proper cooling, no?
Yes, sir. Only in 450 ml bottle we are feeling we are getting such. Not in 200 ml or in 100 ml.
Okay, so then it could be something due to something related to the quality of that bottle.
Okay.
Otherwise, it should have happened in the other 200 ml also, no? If something is related
but,
with the...
not in any other Thaila also in 450. When Dhanvantaram Colombo, Sajrati Colombo and all, we are not seeing such thing.
If the problem lies in the what we say in the kind of in the product complaint, there should have been presented in entire batch or this in the 200 ml or 100 ml, whatever it may be.
Yeah, but we are not seeing it in 200 ml or 100 ml. Only in 450 ml of Pindathyla only we are seeing. Not in any other Colombo or
Is this
Thailam.
a frequent complaint or occasional one?
Okay.
Okay, is it frequent?
Yeah, when we fill for 2 times also, it was similarly we are finding in that that became suspicious, but we are not seeing it in 200 ml or in 100 ml bottles, and not even in other bottles.
Is there a ridge in the bottom of that bottle?
Sir?
Ridge. Is there a ridge in the bottom of the bottle?
No.
Okay.
The bottle is a plain bottle which we have.
There is no ridge.
No ridges. We are not we don't have that ridges bottle.
Okay. Usually in the bottles with ridge, it's quite common, but that need to be analyzed completely.
Okay, sir. And one more thing, while preparing the Dashamula Kashaya, most of the time we are getting the output very less.
Only with Dashamula? Up to what?
Dashamula Kashayam. Most of the time.
Up to what degree you boil that?
And usual Kashayam preparation method only we are preparing, and the temperature is also maintained. But still, every time we are getting the output very less.
Probably
even if we are putting it for a 50 liter Dashmulam Kashayam, we will getting hardly 30 liter or something, or we will be getting. Most of the time, the output will be very reduced.
What about the thickness? Is it high, thick?
No, Brix value is normal.
Viscosity is normal.
Viscosity is normal.
Okay. Do you add this? How do you this boil it? Is it a fractional one, or stage by stage, or continuous boiling?
Continuous boiling in moderate temperature.
Moderate temperature. The main issue happens with the excessive sedimentation of starch.
Okay.
So have you seen this starch in the bottom of the tumbler? No, not in the bottle, in the bottom of the what we say, the vessel.
Yes, sir. We have seen that, but still, even the manufacturing manufacturing unit supervisor, they also tell every time we are getting in less quantity only.
You can do one thing. I think the excessive sedimentation, the excessive extraction is due to the sedimentation of starch happens.
Okay.
Starch is being extracted, and so probably the starch comes out from the raw materials when it boils more than 80 degree, 80 or 100 degree, beyond 100 degree. So I think if possible, you can just fix the temperature up to 80 degree.
Okay, sir.
That will be a beneficial. Below the 80 degree, or if you boil in the 80 up to 80 degree for a longer time also, the starch extraction will not be there.
Okay.
And the the pharmacological efficacy also will not be that much hampered, as the starch has nothing to do with the pharmacological activity. So all the other active principle, the ingredients, the primary metabolites will be there in the even if it is being boiled up to the prescribed amount. Except the starch, everything will be there. So always you can just try like this. You can just boil up to 80 degree for a longer time.
Okay,
And
sir.
you probably that will help you.
Okay, sir. Thank you.
Any further queries, please? Okay, now I welcome Dr. Sharad for a word of gratitude.
Thank you, sir. Thank you so much for very informative section. And also, I hope this section is very informative for both Ayurveda doctors and also for those persons who are having manufacturers units. Thank you. Thank you, sir, very much.
Thank you.
Shall I take
Thank
it?
you, Doctor. I would like to take this opportunity to thank everyone who made this meeting possible. And thank you, Dr. Jithesh, sir, for providing such an informative session. And thanking all the participants, especially those participants who are with us from the very first session of the series. And we expect your cooperation and participation in the upcoming sessions as well. And thanks to the management and technical team of Care Kerala. Once again, I extend my sincere thanks on behalf of Care Kerala Limited. Thank you so much. Have a pleasant night ahead. Thank you, sir.